Hi, I'm Dr. Twaseem and welcome to my Derm-classes. So today's lecture is one of the requested topics, which is skin and gut microbiome. So this is one of the most researched topic nowadays, which has both review articles and research studies on this topic. So I have chosen a couple of them to give a review on this topic. So my basic intention is give you a crux on how this actually works so that you can apply this basic knowledge into multiple areas and then dive in deeper if required. So let's get started. So first, about what are the things that are common between skin and gut. As you know, both of these are organs that come in contact with the environment very first. And these both are richly innervated and vascularized. On top of that, they both have neuroendocrine roles, and the recent studies shows that there is a tight connection between skin and gut. There is a bidirectional connection, which we will delve into details in later discussions. So first, about what are the so coming to what actually constitutes the gut microbiota. So this is mainly constituted by not just bacteria, but viruses, fungi, protozoa, etc. And the studies show that the number of these microbiota is 10 times more than the host cells. Similarly, even the DNA is 10 times more than the human DNA, which means we harbor a lot of microbiota, even more than our own cells. So coming to what is the role of this microbiome. So the microbiome has two primary roles. One is the metabolic role and the next one is the immune role. So first coming to the metabolic role, it basically helps in digestion of complex polysaccharides, whereby it also helps in production of certain vitamins such as vitamin K. And so the next role is basically so coming to the second one, which is the immune benefit, so which is basically two of the responses. So one is immune tolerance of the antigens, which are basically dietary antigens or environmental antigens, which doesn't require a immune defense. So the body needs to recognize that as non-harmful antigens. So the gut microbiota helps do that. And the second one is the protection against the pathogens. So we will delve in detail about how each of these is done. So this is one of the pictures where you can identify there is pathogen associated molecular proteins that's present in viruses, in bacteria, which is presented to, which is recognized by the receptors on the host cells, such as macrophages, neutrophils, NK cells, etc. And they actually mount an immune response. So this is how naturally the process happens. So in immune tolerance, what happens is that certain antigens of the gut microbiota is being recognized by the pathogen pattern recognition receptors on the immune cells. So which is like peptidoglycans on the microbes, which is recognized by the toll-like receptors of the host cells. And as an end result of this, what happens is that there is production of immunoglobulin A, there is homeostasis between the T cells. So generally the T cell subsets are divided into two. One is the effector cells, the other one is the regulatory cells. So the homeostasis happens when in a normal case there is more of regulatory cells than the effector cells. So in instances where there is an excess of effector cells is where there is more inflammations. So the body needs to control the balance between the effector cells and the regulatory cells. So what the gut microbiota helps to do is that they helps in a homeostasis between these two subsets of T cells, whereby they increase the T regulatory cells than the T effector cells. So this is actually a crux of what actually happens in an immune tolerance. And then coming to the epithelial barrier, how it is being protected. So there are two mechanisms again. So one is they do directly bind to the intestinal epithelial cells, whereby which they prevent the adhesion by the pathogenic microbes. And the second one is that these gut microbiota basically produce certain proteins that actually provide immune benefit to the host cells. So for example, the lactobacillus, they secrete P450, which actually helps to maintain the epithelial barrier. And second one is E. coli, which is actually contains flagella, which produce beta defensin 2 in the epithelial cells. So they actually provide a immune protection. So this is the mechanism by which they actually helps to provide a barrier function. So this again is a part of providing immunity to us. So let's take a look at this image. As you can see, the gut microbiota is actually helping to maintain a normal healthy gut, where they produce a short chain fatty acids, vitamin D. They help to maintain the homeostasis between the T regulatory cells and the effector cells, thus producing an anti-inflammatory environment. And they also help to maintain the gut intestinal barrier integrity. So it was that in 2014 that they started finding out that the gut microbiota had a distant effect, whereby the gut microbiota influenced not just in colitis but in arthritis and allergy, and they started identifying in various diseases that there is a dysbiosis that they find. So this was basically identified because of the imbalance between the T regulatory cells and effector cells, whereby the effector cells were more than the regulatory cells. So this started to be identified as one of the major causes for the auto-inflammatory diseases. So this is again a pictorial representation where you can see the gut, the probiotics, they interacted with the macrophages and the dendritic cells, which further influenced the immunity, and they help to maintain the brain, the skin, and the lung homeostasis. So by having an effect on the immunity. So coming to the skin, so how a gut microbiome helps to maintain the skin homeostasis. As we know, the skin has primary function in protection, in preventing water evaporation or water retention, and also in maintaining a temperature regulation. So this happens when there is a homeostasis. So the gut microbiota have an influence on the immunity, which further influence this homeostasis. Is being found out in a lot of studies. So let's take an example. So gut microbiota and metabolites like retinoic acid, polysaccharide A, which is produced by bacteria Aspergillus, Faecalibacterium, and Clostridium clusters, have actually found to increase the amount of T regulatory cells, which actually helps in maintaining the homeostasis and providing an anti-inflammatory environment. The next thing that they found out is the effect of short chain fatty acids, which is actually produced by the gut microbiota. They again have an influence on the proliferation, the migration, the cytokine release, the adhesion, etc. So again, a lot of bacteria and their effects. I'm not delving in detail about each of them because that's something that is very self-explanatory. So the bacteria like Propionibacteria, Lactobacillus brevis, and Reuteri have an influence on maintaining the skin homeostasis, according to the studies mostly done on mice. And they also help in maintaining the keratin, help in the keratinization process as well. So coming to what happens in a dysbiosis. So in dysbiosis, as we all know, what happens is that the amount of gut commensal is less and the amount of the pathogenic bacteria is more. So one of those is the segmented filamentous bacteria, and it's found that they promote the accumulation of Th17 and Th1 cells. As we know, Th17 is one of the cytokine, is one of the main mediator for psoriasis. And another thing that's been seen in the dysbiosis is that the intestinal barrier integrity is being disrupted, thereby the bacteria can directly enter and the metabolites can directly into the bloodstream, and they can reach the skin and disrupt the skin homeostasis. So this is again a pictorial representation. I'm including a lot of images so that you will have a solid idea of what this basically is. So let's take a look at the homeostasis, where there is more gut commensals than the pathogens. They're producing antimicrobial peptides, which keep in check the pathogens. They are interacting with the immune cells, whereby they're increasing the T regulatory cells. They're also providing improved gut integrity, and they are producing an anti-inflammatory cytokine response. And the second one, where there is dysbiosis and there is more of pathogens and less of commensals, you can see the pathogens have even entered into the bloodstream because there is a disrupted gut integrity, and there is also more inflammation. So there's a pro-inflammatory cytokine increase. So the pathogens like Clostridium difficile, they produce metabolites like phenol and p-cresol. They can accumulate in the skin and they can impair the differentiation. So again, they can also affect the effector cells, as we've discussed already. So they basically impact on the integrity and the immunity. These are the two main things that you have to keep in mind. So this is again a pictorial representation where you have more pathogens. So there is more inflammation, there is a leaky gut because of the disrupted barrier, and there is more of inflammation. This is further causing acne, this is further causing psoriasis, this is further causing atopic dermatitis. Coming to what is the role of microbiota in allostasis. So what is first of all in allostasis? Allostasis is a scenario where the disrupted homeostasis is being reconstructed or reconstituted. So, so that again is influenced by the microbiota. So we have various bacteria like Lactobacillus helveticus, Paracasey, and even Reuteri. So again, the mechanisms I have described in the slide. If you want, you can pause for a moment and take a look at that. Anyway, these microbes is helping in maintaining the homeostasis by increasing the hydration, keeping the inflammation under check. Again, these are the various other mechanisms by which different gut microbiome is helping in maintaining the homeostasis. So now let's delve into the dysbiosis and its effect on skin diseases, mainly acne, atopic dermatitis, psoriasis, and even disorders like rosacea, in urticaria, in hidradenitis suppurativa, in alopecia areata, and various other diseases. I will only be dealing with the main. So let's delve into acne vulgaris. So we know that acne is basically caused because of many reasons, which include sebum oversecretion, keratinocyte desquamation and obstruction of the duct, and also by the Propionibacterium acne. So what's been seen is that due to the high glycemic index foods, which increase insulin-like growth factors, which further causes influence on the mTOR pathway, this causes the increased sebaceous gland activation and resulting in the acne. So this is one of the schematic diagram which shows that even the mTOR pathway also has an influence on the intestinal barrier. So this is basically a diagram that shows an influence of even the psychological factors on acne. So when there is a psychological stress that can actually cause increase in the substance P nerve fibers in the skin that can directly cause acne. Similarly, they can also increase its influence, similar influence in the gut and cause a dysbiosis. And on top of that, as mentioned already, the junk foods can cause insulin-like growth factors more in the gut, again causing dysbiosis. See, all these factors is contributing to the formation of acne. So another thing is when you have a psychological stress, there is low levels of acidity, which increases the overgrowth of bacteria, and which again causes certain accumulation of metabolites as there is a defective absorption, which further causes a psychological stress. So all these multi-factors occurs in a cyclical manner due to an interdependence. So nothing is separate, everything comes together to influence the pathogenesis of the diseases. Next is about the atopic dermatitis. As we know, it's basically a multifactorial. It can be filaggrin deficiency compounding with the, an impaired immune mechanism that results in atopic dermatitis, where there is more Th2 in the acute phase and more Th1 response in the chronic phase. So one of the theory is the dietary microbiome theory to explain the atopic dermatitis, which mentions due to the Western diet, there is more fat and less dietary fibers, which changes the gut microbiome. And this also, due to which there is a defective production of metabolites like short chain fatty acids, which produces leaky gut and which basically disrupts the immune homeostasis, which can produce an exaggerate response to the environmental antigens and the dietary antigens. Similarly, in psoriasis, as we all know, there is an exaggerated Th17 cells and the cytokines involved. This even has a role in IBD, the same cytokines. So certain studies of psoriasis have found that there is this microbes in the blood, which shows an influence of the leaky gut. So these are all the factors which shows that the dysbiosis can be a cause for these inflammatory diseases. Similarly, one of the studies in Korean patients about the gut microbiome and rosacea have found that treatment with rifaximin has improved the rosacea. Basically means it's probably because of the bacterial overgrowth. Similarly, there's been other bacteria that been found to influence diseases like hidradenitis and chronic suppurative urticaria. So this is again one of the tables from the review articles, which basically mentions what are the different probiotic strains which has actually helped to treat some of the diseases. So you can have a look at them as well. So finally, what I would like to tell you as a take-home message is, in a lot of inflammatory diseases, we should not just treat the inflammatory disease, rather we can also aim to target the gut dysbiosis. So probably we can go for a holistic approach. So that is the primary takeaway about the gut and the gut microbiome and the skin diseases. And as I've already mentioned, this is just a crux of it. So in case you want a very detailed, you can delve into any of the review articles, or you can also delve into the individual researches that's happening. And this is just happening in all diseases of skin, not just the ones that I've mentioned. Some's been proven in humans, some just in the mice studies and not in humans. So anyway, everything is under research, so we can expect a very solid evidence of different microbes causing different diseases, and what are the microbes that can be used to treat different diseases. And I think this has been incorporated into the practice nowadays. So we need to have a very good knowledge about the different gut microbiomes and their. So these are some of my references. Feel free to delve into the rest of them. And I hope my lecture has helped you in clarifying the concepts. Let me know in case you have any doubts. And thank you so much for listening.